Okay, good afternoon, everyone. It's my pleasure to welcome you to this today's symposium on HMOs, microbiome-related benefits and their potential beyond immunity. My name is Hania Szajewska. I'm professor and chair of the Department of Pediatrics at the Medical University of Warsaw. It will be my privilege to chair this symposium. Here are my faculty disclosures, and allow me to start this symposium organized jointly by Nestlé Nutrition Institute and Wyeth Nutrition Science Center with a very short introduction to today's topic. Human milk oligosaccharides, the third most abundant solid component in breast milk. And you know that it's known for years, but in a way it's being rediscovered. Known for years because the first observations are coming from the 19th century, but we are really observing a growing interest recently. And you may ask yourself why there is such a growing interest in human milk oligosaccharides, in HMOs. And there are many reasons, but I think the two of those are really important. First, advanced techniques allow us for detailed analysis of HMO's structure and function. And second, which is equally important, is that modern biotechnology enables nowadays large-scale production of selected HMOs to be added to infant formulas. And you can already understand now that we have these human milk oligosaccharides which originate from breast milk and those which are biotechnologically produced and being added to infant formulas. There is a lot of discussions, at least within ESPGHAN, how to differentiate those two when it comes to terms, to the terminology. And some of these terms which are being considered are presented on this table, on this slide. And after many discussions, we came to the conclusion to use a terminology which is also used in some of the EFSA document, European Food Safety Authorities document, human identical milk oligosaccharides, HMO. It's not perfect, but it's really important to differentiate those two. We are at the ESPGHAN meeting, and I'm sure that many of you already attended some of the sessions that ESPGHAN Special Interest Group on Gut Microbiota and Modification presented a 6-paper series on biotics being added to infant formula, including among those biotics added to infant formula were human identical milk oligosaccharides. In all our documents, and as you can see, all technical reviews are already published or accepted for publication. In all our documents, and also in the summary recommendation paper, we focus on clinical outcomes. And one of the frequently asked questions, and perhaps some of you may ask it also today, is why ESPGHAN Special Interest Group on Gut Microbiota and Modification did not include microbiota data. And the answer is that changes in the microbiota alone without demonstrated health benefits are not, in our opinion, sufficient to support recommendations. However, this does not mean that such data are unimportant. On the contrary, and this meeting, this symposium today, will really focus mainly, not only, on those HMOs and the effects on microbiota. Once again, the topic is HMOs, microbiome-related benefits, and their potential beyond immunity. We will have 2 presentations, which will be followed by a panel discussion. Our presenters today are Professor Anne Salmen. She is, and allow me now to read, principal investigator and adjunct professor at the University of Helsinki, Finland, and director and Human Microbiome Research Program, Faculty of Medicine, University of Helsinki, Finland, among many other of her functions. And the topic of her presentation will be From Genotype to Gut, the fascinating role of HMOs of infant microbiota on health. And the second speaker today will be Dr. Norbert Sprenger, who is a senior research and development expert from Nestlé Nutrition Institute of Health Sciences, Lausanne, Switzerland. And he will talk about human milk-inspired biotics, building blocks for health and beyond immunity. So we will start with the first presentation from Professor Anne Salomon, and this will be followed by the second presentation by Norbert. Anne, the floor is yours. Thank you very much for your attention.
Good afternoon, everyone. And thanks for the organizers for giving me the opportunity to talk today about the breast milk-based human milk oligosaccharides in relation to infant microbiota and health. Indeed, my lab is just a couple of kilometers from here. I really hope you have enjoyed Helsinki during this conference. So while we are all excited about the biotech side of the HMOs, that you've been hearing several presentations throughout this conference as well. And it, of course, gives us wonderful opportunities to look for causality and mechanism of action. We have to keep in mind that there is a lot to learn still also from the observational studies, because basically only looking on how the breast milk HMOs interact with the gut microbiota and associate to gut health is basically how we can open the biological blueprint. In other terms, to give us the nature's recipe, how to make the optimal HMO blend we all know is supporting optimally infant development in breastfed infants. Of course, we are faced with tremendous complexity, but we have to acknowledge the variability of HMOs across women in nature, not only between populations, but also in terms of lactation of the child age, and in consideration of also some subgroups that we start to identify, and I will be elaborating all of this in my talk. We are inherently limited to associations when we look at the breast milk HMOs, but we can still get very useful ideas whether the abundance of HMOs actually equals to their biological activity, and we can also learn a lot from the synergies and specifics of between HMOs. So in the breast milk HMOs, I will in my talk address these three main knowledge gaps. First of all, what do we know about the factors that are explaining this big natural variation we see in breast milk HMOs? Which are those HMOs that actually associate to gut microbiota variation in infants that we know is so important for the programming of the health of the infants, and in which specific ways? And do actually the differences in HMOs that we see across women, do these differences, are they relevant actually for gut microbiota and health? Or in other terms, might all HMOs actually be equally good? And I will give you some learnings from the recent studies. So as said already, I will completely focus on studies that have been conducted on HMOs, gut microbiota. Few studies also have health outcomes available done in breast milk, while there are a lot of publications. But what I do in my presentation, I'll summarize for you the learnings from 3 large, very recent studies, because actually you do need some power to do HMO research in human milk for reasons you will hear just in a second. So the summary of my presentation will be based on the breast milk HMOs of roughly 1,000 European women. Majority of them come from the Dutch cohorts, a koala cohort, and a very big lifelines next cohort, where the results are actually not yet officially published, but there is a PhD thesis you will find online. And then our own study, which is actually the largest study, observational study published so far, where all the 3, so the HMO, breast milk HMOs, infant gut microbiota, and including health outcomes were available. So as said, altogether these cover roughly 1,000 women, in total 2,000 breast milk samples. So our local cohort, health and early life microbiota birth cohort, it's a cohort of 1,000 children and their parents, and we've recruited this cohort together with Willem de Vos, Kaja-Leena Kolho, several years ago. And the focus of our research here has been really to understand the impact of various early exposures, including diet and HMOs, on gut microbiota development and importantly, on the well-being. And those health outcomes we can actually measure in a population where the neonates were born healthy. And we have collected over 10,000 fecal samples, and by now we've published dozen plus papers, including both clinical questions by Kaja-Leena Kolho, featured here on the left, as well as the microbiota-focused papers, including the Gut Microbiota Wellbeing Index paper that has been featured a couple of times already in this conference. So in my talk, I will summarize our findings from the HMO study. This was a PhD thesis work by Dolvin Mataru, and the results have been published in eBiomedicine. So we had 350 breast milk samples at age of 3 months. We had several samples from fecal samples from infant up to 1 year, and we had several health outcomes until 2 years of age. And the HMO analysis was done in collaboration with Glycom, that time DSM. Before I go there, I just have to give you a bit of background. So you probably all know not all breast milk indeed is not the same. And there are 2 main factors that we know for very long to affect the natural variation of HMOs. If you first look on the right, this gives you an idea of the temporal development. Most of the HMOs are in highest concentration in colostrum and in neonatal states, and then the concentration decrease. Of course, the volume of the milk intake increases by the growing baby. But there is something which actually affects much, much more the concentrations of the HMOs in breast milk, and this is the maternal genotype of 2 genes, the polymorphisms in fucosyltransferase 2 and 3, also referred to as secretor and Lewis genotype. And as you see here on the right, only secretor mothers can produce 2-fucosyl-lactose, which is, if you take a mean over Western populations, it is the most abundant HMO in breast milk, here the pale green, but it's only present in secretor plus milk. So that the non-secretor mothers have, you can see that the total concentration of HMOs is lower. They are completely lacking 2FL, even though the total amounts is to some extent compensated by 3FL. And you can also see that even within these genotypes, there is also quite some variation between the women, and based on these two genotypes, it's very common that all the current studies basically use these 4 milk groups in their analysis. And roughly 20% of individuals in Western populations are non-secretors. However, there is a huge geographic and ethnic variation, ranging from the non-secretors are basically absent in Latin America, while up to 40% of the population have this genotype in certain parts of Africa. It's partially related to the pathogen load, or largely related, because we can discuss in the QA what implications these genotypes have for our resistance for pathogens. But this is just to keep you in mind that even if you would study exclusively breastfed infants in different populations, the HMO composition and the nutritional content, if you like, is quite different. So in our study, we utilized the rich metadata, and we screened for several dozens of different factors, ranging from technical to different maternal or perinatal variables, to answer the question that which are the variables, factors that, apart from the genotype, mother's genotype, affect the HMO variation. And one that really stood out was the sampling season. So in our Finnish data, we saw that basically almost all HMOs were highest in autumn and lowest in spring. We could not, in the analysis, trace it to any variable, but our strong hypothesis is it relates to diet. Because in Finland, the diet is quite seasonal. The fresh vegetables we eat much more them over the, when we can get them locally. In our cohort, we did not find basically links to maternal anthropometry or other variables, which some other cohorts have identified. And I have to mention that the seasonal effect, as well as the parity, have been reported earlier, for example, in the large Canadian child cohort. And in that perspective, it was quite interesting that this big Dutch cohort, this Lifeline's next cohort of 700 mothers, did not find season and parity in Dutch women to be important in explaining HMO variation. You can see maybe that actually quite few of the many tested variables actually stayed significant after correcting for multiple comparisons, but they did identify maternal ABO blood group prior smoking and subclinical mastitis explaining HMO variation. So we are learning about the underlying reasons to explain the HMO variation. Then the big question, how does this HMO variation, and in this case first, the mother secretor status, in other words, the presence of the major HMO 2FL in the breast milk, how does that relate to the community level infant gut microbiota variation? And when I talk about community level, I refer to these beta diversity plots where you should see the groups separating if there was a clear effect. And if you look our data, the blue ones and the mustard colors are the secretors, while the black ones and the orange ones are non-secretors, and you can see they are all scattered. In other words, the FUT2, presence of FUT2 in the breast milk in exclusively breastfed infants does not explain infant gut microbiota variation. And it was exactly the same conclusion that the Dutch study did. In our case, because we had metagenome data, we actually also looked for the enzymatic activities, and somewhat to our surprise, we also did not see that the cases, the carbohydrate active enzymes, also did not differ according to the content of the milk. And this same result was actually already published earlier from this koala cohort. So you see this dramatic, on the upper row, you see this dramatic variation in the HMO composition, so that the orange one, the 2FL, which is very present in the secretor mothers, completely lacking in non-secretors. Despite this variation, there is very little variation in the gut microbiota composition, because the top row, you can't read it probably, this is for bifidobacteria, and you see these warm colors indicating high abundance, also in those infants fed with non-secretor milk, while they are absent in the infants fed with normal formula. So when we look at the individual HMOs, the individual taxa in the infant microbiota, we start to see some differences, and this is the same for all three big cohorts. However, the take-home message, for sake of time, I don't go into details. The take-home message is that that the key HMO consumers, we know bacteroides and especially bifidobacteria, they are typically not showing association to, especially for the major HMO. And we started to ask ourselves, is this really true? Or is there something that might mask the effect? So what we did then, we stratified our data according to the birth mode, with the rationale that, because the birth mode is the factor that is most affecting infant gut microbiota variation. And we start to see the associations then, meaning that in breastfed infants, and I think this is applicable also for supplemented formula, the HMO infant microbiota associations are actually highly context, in this case, birth mode specific. And we see the absolutely highest number of associations in babies who were born by C-section, where we basically see this, what we were actually anticipated when we started the study, that all the positive associations are with bifidobacteria, and the negative associations are with pathogens or pathobions. For example, that the higher the amount of total HMOs or total neutral HMOs, the lower the abundance of Klebsiella in the infant gut. Then finally, we come to the health. So the few cohorts that have also been able to, that have collected health outcomes, the question is that whether the maternal secretor status or breast milk HMO, once again in exclusively breastfed infants, whether that associates to child health outcomes. And the question is that in most of these recent large-scale European studies, the answer is actually no. In our case, we had the health data at until 2 years. For Lifelines Next, it was a very brief, short follow-up, only until 6 months. And the child study did not have a microbiota for this high amount of babies, but they could show that when they reanalyzed their data, stratifying the data for the secretor status, they could show that the breastfeeding by non-secretor mothers was equally protective against overweight and asthma as non-secretors. And there are also some positive studies I have to mention definitely. For example, there is this quite big Finnish cohort from Western Finland which showed an association between 2FL and mother secretory status for height and weight. And this morning, it was also highlighted, the paper highlighted by Professor Donovan, an earlier paper from the next speaker, and Mikael Kuitunen, where the placebo group of a clinical trial showed that in C-section babies, also the infants of secretory mothers had less allergies. So how to interpret all this data? To conclude, so for the sake of time, I just go for basically to the lack of association. So to recap, we saw no or very limited associations of the mother secretory status or the HMO composition in breast milk, either to infant gut microbiota composition or health outcomes. And the clinical and practical implication is that as long as the baby is breastfed, it seems that the protective effect is largely independent of the HMO composition. And while this kind of might sound counterintuitive, if you think about it from the evolutionary perspective, all critically important biological systems are backed up with redundancy. It's kind of an insurance system. And my interpretation of the data is that in HMO infant health interactions, there is two-level redundancy. We know that, first of all, that's within the HMOs, that it seems, even though they are structurally different, there is certain amount of overlapping functions. And we also know that the key bifidobacteria, for example, that really are fully dependent on the availability of HMOs in the neonatal gut, they can use more than just one or few HMOs. And this is very important for providing a kind of adaptability so that not all is played by one card. And just my final conclusion is that I think we are moving towards understanding the context specificity of these dependencies and interactions. The one we identified that the C-section babies had completely different and much stronger associations with the HMOs and the milk and the infant microbiota indicate that we should really zoom into the subgroups. And this, I think, provides a rationale for targeted nutritional support in some of these subpopulations. And I'm looking forward for the Q&A to discuss more about this. Very much thanks for your attention.
Thank you very much, Anne. And yes, we will have Q&A session at the end. I hope that Professor Salonen was able to convince you on the importance of human milk oligosaccharides and the effects on gut microbiota, and also the importance of breastfeeding, therefore. And as I already announced, the next speaker is Dr. Norbert Springer, and the title of his presentation is Human Milk-Inspired Biotics, Building Blocks for Health Beyond Immunity. Norbert, the floor is yours.
Thank you very much for the introduction and having me here today. I tried to get the pointer, but I don't know how to do that. The mouse seems to be blocked. But okay, no pointer. I will just talk. So I will talk about biotics inspired from human milk, and I will start, to show you something maybe as a conceptual framework of how we study actually these different biotics. But before going there, I have also my disclosure. So I'm an employee from the Société des Produits Nestlé in Switzerland. That's how we are called, at the Nestlé Institute of Health Sciences, again in Lausanne, Switzerland. So my outline of the talk today is a little bit of a context. So you saw already a lot of context from Anne. And I will then go more to HMO promising biotics as part of this exercise we do by learning from human milk, and then how we bring that into a formula to improve if an infant cannot be breastfed. And then the last point is first insights from HMO with Bifinfantis as a new probiotic. And here I will show you some data from the recent trials that we did, where I show you some unpublished data, so please keep that in mind. We are publishing them now with abstracts here. We published some of them as abstracts before, and we're writing up the papers as I speak. So I want to start with this more conceptual framework of a pyramid. So we have to consider like a nutrition pyramid. There is a base for everything, and there's a foundation that is very important. And you can see there, what I put is the nutrition in a handshake with the gut microbiome. So I think this is really a very, very important foundation, especially in infancy, where you start to learn to live with your microbes that come in as you are born, and then basically they develop with you, and you need to take care of that very, very carefully, because that handshake is changing with time. And we need to consider this in a time frame, and everything has to be time adapted, so time appropriate. So very, very important concept, and I think you had maybe also heard Willem de Vos yesterday talking about this. We see data coming in from different big cohorts showing you the right pace of development and how we learn how important this part is. So now I show the next level, which is happening more or less in the infant. It's the metabolism, it's the digestion, it's the absorption of all the nutrients. And then, of course, the immune system as a kind of a garant of getting it right also in the infant as the infant grows. And as a first entry point, obviously the gut and the gut health with all the different mechanisms to make sure you don't get allergic, you can tolerate your new food, you can develop no inflammatory condition and so on, is a very, very important point. So that's my first point. Now we said also we go beyond immunity, and that's the upper part. So we have physical growth, and everything ended up here in healthy infant development. And we have also the skill development growth that I put there. And so there are different elements that we can study, different parts that all contribute as a unity to eventually have healthy development. So I show you this because I think this is a framework to keep in mind when we talk about nutrition. And it's a framework also when we talk about how we make associations from observations plausible. Because they're usually not a causation, they're just an association. So we need to be extremely mindful on this. And basically, this framework can help, because then we can see, okay, what could be the link to some of the observations? Now, before I come to some of the associations, I also want to give you a little bit of a snapshot on some of the risk factors. Many of them we discussed already at this conference in many different sessions. So you see the risk factors for appropriate gut microbiome development. So this is the headline, and you see the feeding mode is the first one. So, for example, formula feeding compared to human milk feeding or breastfeeding is something that changes how the microbiome develops. Antibiotic exposure, very obvious. Delivery mode, it's another very important point that comes often together with antibiotics, even perinatal, and this was also shown nicely by Anne and her team, how that affects actually the C-section effects that we see. And then family size, something that maybe we don't consider so much, but again, I'm really fascinated by this Helsinki cohort where we could nicely show that having siblings is actually changing how your early life bifidobacteria develop. So there is some kind of a mechanism how you learn to live with your bifidobacteria when you have a brother or a sister, and maybe comes through the mother. Maybe we can discuss it later, but I found this extremely fascinating. And as you know, societies change how family structure is. We have, smaller families in one house, not big families with all your brothers and sisters or cousins and so on. Excessive hygiene, I don't want to talk more about this, but I think that is also a little bit of something to be discussed. And environmental exposure of other bugs that come in or don't come in as transitory microbes. But then I put there 3 buckets in the middle that you can see. So a human milk adapted HMO utilizing bifidobacteria population was shown now repeatedly in many studies, observational mainly, to be important, especially for immune health later. These are the things that are first tested and studied. And then what I mentioned also is this age-appropriate pace or speed of development. And that is not only in the, let's say, exclusive breast milk feeding period, but also after that, after 6 months, when you start to diversify your diet, you need to also make sure nutrition helps you to diversify your microbiome, because otherwise you end up again in a problem. And then the precautious acquisition of the taxa comes a bit with this pace, because you have other taxa or bacteria that could come in that you don't want at the early phase, but they're very beneficial later maybe. And so that's another point. I will show you some data on this. And then eventually I just put there a picture on this more pro-inflammatory condition, especially with a disbalance that remains with a higher Th2, T helper cell 2 and T helper cell 17 population compared to more balanced with the T helper cell 1, which could be a risk for allergies later in life. And we actually showed that part also in animal models. Now, just coming very quickly to our effort also to learn from human milk. So we have a big initiative which is called LIFE. And basically there we have a collection of many, many studies on human milk-fed infants that we study very broadly. We also do a lot of validated method development to study human milk composition from all kinds of components. And we distribute that by publishing or by making sure others can also access to this. So we have some method even implemented in CROs that can be used by anyone who has no facility to do HMO research. They could just pay and basically do the measurements there. And then you see just on the map, we're more present, let's say, in the northern hemisphere, a little less, unfortunately, in the global south. But that is something I think we need to change in the future. And then I come just to a few examples here to show you what we study in terms of that pyramid. So I show you especially on the left side more on the physical growth part, that we also look at bone quality, for example. And Nicolas, my colleague, he has a poster that you can listen tomorrow and discuss with him more in detail. And what I wanted to show you here is that we also try then to come from the observations to causality by doing actually also preclinical models where we can ask more specific questions and also dissect the animal basically to understand mechanism of action. So we try to confirm it through that. And on the other side, you can see examples of another cohort where we do again this approach of association, bringing it more to causality by doing mediation analysis. So mediation was mentioned also through microbiome as a factor that we can do. What we did here is to look at the mediation through another marker of brain development, which is myelination. And we had very interesting findings here that confirmed somehow also the models that we used on the mouse, where we have knockout mice that do not produce the cellulated molecules in the milk, and then we cross-feed to wild-type pups, and we study basically their brain development and behavior later. And this is a package of data that really indicates there is something. We don't know exactly the mechanism, but there's something behind all those molecules. And if I just summarize that very briefly for you here, so the different manufactured HMOs or HIMOs, they're illustrated here in those symbols. And you see with the red triangle, it's all the fucosylated ones. Without the red triangle, you see the neutral, non-fucosylated. And then with the diamond, you see the cellulated ones. And you see there's buckets of different core benefits that were shown or proposed by the different associations combined usually with preclinical models. So this gives us a little bit of a summary of the promise these molecules can give as a biotic. Now, what is most interesting for you, and I hope I have you all with me still, is actually the clinical findings from trials where we can also study this in a different matrix now, in an infant formula, and look at those manufactured HMOs basically in randomized controlled trials. So I thought I would just summarize with two basically review articles. One is from the Espegan committee, the other one is from an independent group of researchers. And they summarized basically what happened with all the different trials. And you can see here the highlight, the key findings, key learnings from all that summarizing of the different intervention trials. Is they're safe, they're well tolerated in adults and in infants. And in infants, there's generally seen a shift towards observed or measurements that were observed in breastfed infants. This concerns mainly microbiota outcomes, but also some others, and I will show you some of them. And we don't know yet exactly what to conclude in terms of infections, for example, or any other outcome. I think there is really a need for then dedicated research specifically for those outcomes. So now let's dive into the microbiome a little bit more in detail. And what I show you here is two studies. So one is an observational study that basically observed in different populations from a non-industrialized population to a transitional population to industrialized populations, how the bifidobacteria populations shift. And this is also something we heard yesterday from William de Vos, that there seems to be a decrease in those HMO metabolizing species like Bifidobacterium longum subspecies infantis. And that is nicely shown here, that you have this dominance in the pink one of Bif infantis, and then you diversify the different Bifidobacteria. Some can metabolize human milk oligosaccharides, some cannot. But the master metabolizer is that group of the Bif infantis. And then what you see in the other part of the left figure is basically the metabolization of human milk oligosaccharides that also is decreased as you industrialize a certain area or a region. And then on the right side, you can see something from our intervention trial of infant formula fed with 2 HMOs in this case. And what we observed when we studied the microbiome is that we have basically 2 main community types in early life at 3 months. So one is the red group, and that is the most dominant one in the control formula fed infants. And then there's the green group, which is the most dominant group of microbiota in the breastfed infants. And you can see when we actually supplemented the infant formula with 2 HMOs, we get more or less half of the infants green and the other half stays red. So it's like we have a non-responder phenotype that we see in the gut microbiome. And basically what we could show is that indeed this goes together with the loss of that HMO utilization capacity in the gut microbiome. And this led us actually to revisit the whole concept of the HMO feeding, together with also literature data, and to propose that we need to invest into a new probiotic. And so what we did is we basically looked at 17 different strains. We basically characterized them in detail. We did our criteria how to select them, and basically in the end we selected this B. longum subspecies infantis LMG 11588. So this is our new strain that I'll show you then the intervention trials. It is safe, it does not harbor any antibiotic resistance genes, so this was one of the key criteria that we also put up. It can metabolize a wide range of HMOs. It's actually focusing a lot on the fucosylated and a little less on the neutral non-fucosylated, which we identified to be more broadly used actually by bifidobacteria. So thereby we also don't disturb maybe the natural bifidobacteria that are present, and we can still feed them with the more neutral ones. So that was another reflection we had. And then it shows the synergy with the 6 HMO blend that we studied here in detail. And it resulted actually in a synergistic increase of metabolization, of the outcomes from the metabolization in terms of metabolites that are beneficial generally for the immune health. And we could also show in an animal model that there's a synergistic benefit when we put the 2 together compared to each ingredient individually in terms of respiratory infection in the mouse model, followed by a subsequent allergy prevention, that we induced an airway allergy there. It integrates well into the gut microbiome, and it does not replace in all our studies any of the inherent present B. infantis strains. So we had a very sophisticated analysis there, and I invite you to a poster that is still hanging around, where you can look at the details there, or you can ask me later. So now let's come to the trials. So I will show you data from 2 trials. The first trial, trial 1, is a 6 HMO trial with a blend that we adapted from stage 1 to stage 2 formula to stage 3 formula by adapting the concentration and also the proportion between the different HMOs. So mainly we increase the 3FL concentration while we decrease, for example, 2FL concentration, something that is typically seen in human milk. And then we have the others also adapted in a similar way. And we combined it with 2 probiotics, in this case our B. infantis and the B. lactis that we also have generally in this type of formulas. And in the second trial, we further enriched basically the HMO blend, in this case were 7 HMOs, with a bovine milk-derived oligosaccharide preparation that we use as a base in these formulas, and we combined it only with the Bif infantis. Now the age-appropriate growth and the tolerance was also shown. I don't show you that here, but please believe me, and it was shown in previous posters and presentations, and we're writing up the manuscripts. So now let's deep dive a bit in the gut microbiome and how it looks like. So I show you here a picture on how we also try to understand the microbiome by basically describing it through age-discriminant bacteria that are at different times important. And basically that shows you, when we take as a reference the breastfed, which is in orange, it gives you a diagonal line. So it can very well predict the infant age, and then it flattens out. So it means, you stabilize now your gut microbiome development. And there are different bacteria involved in that, and now I'm really sorry I cannot point to that. But you can see in the different colors of the blobs, you can see some that are important for later time points and some for early time points when we look at the reference. So early time point is in the dark blue, and I show you there the bifidobacteriums at the species level, at the genus level, just all together. And you can see that, in fact, what happens is that we increase the bifidobacteria in the experimental formula significantly until 6 months. Then overall for bifidobacteria, we don't see the effect anymore in the formula feeding, but we still see individual bifidos like Bifidobacterium bifidum and breve that increased also in stage 2 and stage 3. And we see also diversity of bifidobacteria more close to the breastfed. And then the non-appropriate or too early or precautious bacteria, I show you here an example of Ruminococcus gnathus, which is a mucus forager, so it can also nibble around on your mucus protective barrier, and basically could be a problem maybe for gut barrier function. And we see that we decrease that significantly again with the experimental formula. So as examples of this precautious establishment of taxa. Now we also see that in terms of the short-chain fatty acids, so we have in the experimental formula a slowing down of your butyrate producers, indicating again that we slow down that development, but then we have actually a faster increase towards the stage 2 and stage 3 measurement that we had to basically adapt that pace of development. And you can see that in the middle part of the figure, that goes also together with the modules on the butyrate producing bacteria when we look at the capacity to produce butyrate. That was already higher in the control formula compared to the experimental formula. But later, as you see, then the butyrate is even slightly higher, and we see in other trials also that we can associate actually the early bifidobacteria peak, especially Bifinfantis, to later butyrate producers. So it's like there is a preparation of your gut microbiome through that early bifidobacteria pool that helps you to be in a healthy microbiome also later when it stabilizes. So I don't know how serious the watch is, but I try to speed up. 1 minute, okay, that is serious. So I show you from the second trial also some of those findings where we see even stronger effect. We don't have here a reference arm, we have only the 2 experimental arms, so the control formula in green again, and in blue the experimental formula with the 7 HMOs and the BMOS added plus the Bifinfantis. You can see the bifidobacterium very highly increased, also the infant type bifidobacteria highly increased, and specifically then in Infantis highly increased. And we see again the same signal that the Ruminococcus gnathus was even stronger decreased than before, and other bacteria that also should not be too early, like Egertella lenta that was associated to allergies, also decreased significantly. Now what does this mean in terms of gut health? So we measure different gut health markers. You see them there, calprotectin, alpha-1 antitrypsin as more problematic markers with a gut barrier function or a pro-inflammatory condition. And you can see that we significantly reduce the calprotectin and alpha-1 antitrypsin at different time points with our experimental formula with the synbiotic. And what you see below is the association of these markers with Ruminococcus gnathus specifically. So when it's higher, there is higher of that pro-inflammatory marker or gut barrier marker. And when it's and that is the highest ones in our control formula. And then on the other side, you see the secretory IgA on the beneficial side. Again, you see the Ruminococcus gnathus popping up as something that actually goes with lower IgA produced in the gut, while on the other hand, the bifidobacteria with Infantis breve bifidum actually are increased. Now, just very, very briefly of what we also try to do is to see how we reduce risk factors related to antibiotic use. So what I plotted there, or what we plotted there and what I show you here is the breastfeeding arm with the antibiotic use, especially in stage 2 and 3, where we have more antibiotics, so after 6 months. And that's very similar actually between also then the randomized groups in blue with experimental symbiotic and in green the control. And what we try to do is to understand the effect of the antibiotic exposure at 12 and 15 months on the specific taxa. And I just highlight here the Clostridioides difficile, which we saw in generally decreased with the formula, even in the stage 1, when we have the symbiotic formula. And you can see there that it starts to go up actually in the control formula. So we kind of change something that it can go up. So this is a potential pathogen that we don't want. And then on the other hand, you see also when we look at recovery from antibiotic use, then you can see that we see, for example, this Bacteroides uniformis going up, blooming in the experimental formula, but not in the control formula. And this Bacteroides is actually an important fiber utilizer and an important anti-inflammatory bacterium that is in the gut. So this gives you a snapshot of where we want to go and where we want to study to understand better the link to health outcomes. Now my last little bit in 2 seconds is to show the immune cell profiling also. So we profiled immune cells systemically in a subgroup of voluntary infants who gave also blood at 4 months of age, comparing the symbiotic formula to the control formula and also the breastfed arm. And what you can see and what we observed very nicely here is the reduction in the T helper cell 2 population in the plasma compared also to the control formula. And we had an almost significant reduction of T helper cell 17 cells, which both go in this direction that was predicted when you have bifidobacteria that utilize HMOs, that was predicted in the literature to actually rebalance that disbalance that happened, that is in the early life. And again, this we consider as a risk factor reduction in terms of allergy risks. And this is my summary. So I learned from Hanya many years ago that we cannot memorize more than 3 points at the end. And so I thought I put 3 buckets because I couldn't really decide what 3 points I want to show you. But basically the safety part, I think that is understood. I would focus in the middle part, and I just put again in blue what we changed with our experimental symbiotic formulas in 2 trials, very similar signatures that we see, where we changed the microbiome development speed, and we change in thereby risk factor for specifically for immune-related outcomes later in life. And now we start to learn also what it happens on the immune system with these T helper cells, and from the animal we know that this has a link also to later allergy risk. And last but not least, I want to thank all the participants of the trials, of the different trials, but especially the parents and the caregivers to consent their infants to our trials. And I thank you very much for your attention and looking forward to more discussion.
So we start Q&A session. If you have some questions to the speakers, please, there's a moment. I do have some questions, Norbert, for you. And one of my questions before people start asking is, in your presentation, you use this term gut health. There is a lot of discussion in the literature about gut health, and you use 3 parameters. And perhaps I can question you whether those were the best parameters. A lot of discussion. So please, short answer to this difficult question.
Yeah, this is a very difficult question, and I apologize for giving a very, very broad term to just 3 markers. But I think we need to correct that and maybe have a more accurate nomenclature for this. But
yeah, I would suggest that to avoid using the term gut health, because it's really a lot of discussions, and we are trying also within the ISAP now, we are working on the document, consensus document, not easy. And it's difficult now. Okay, please. There is a question. Please, and introduce yourself.
Okay, it's Polidoro from Brazil. Dr. Norbert, we understand it's very important of HMO because there is in breastfeeding, but we know that the amount of HMOs in breastfeeding is going to decrease during life. So now we are seeing formulas with HMO for more than 6 months old babies, and even in formulas for more than 1 year. I'd like your comments about this. So it's really important to keep giving HMOs even for older child.
I think if we want to follow also recommendation from WHO, it's recommended to keep breastfeeding once you introduce complementary diet after 6 months or not later than 6 months. And in that sense, I think it makes sense to keep the HMOs also in the formula that are going beyond the first year. I want to mention also that if you calculate, with some of the changes in the proportion of human milk by intake and body weight, you can also see that some are actually, since they increase, they stay more constant. So we need to consider this, always in terms of the intake, as you nicely mentioned. But I would definitely think we need to keep them in, because that's also how we recommend, breastfeeding up to 2 years if a mother can still breastfeed. And they would also get the HMOs. They don't disappear from the milk.
We have another question. Please introduce yourself.
Thank you very much both for the very nice talks. Yannick Schünknick from Germany. One question to the clinical trials. You very nicely show there's a change in the composition of the gut microbiota over time, and you also link that to changes in the immune markers. How is that being mediated?
So this we don't know exactly yet. So we believe that some of the mediation comes from those indole lactates or other tryptophan-derived metabolites that are discussed in the literature. So we're still analyzing that. I'm not sure in general that you have just a very pharmacological, one-to-one translation. I think it's really the combination of different components, and we need to be very mindful there, how much we can measure, because it will be a combination most likely. And same for short-chain fatty acids. I think we need to consider them as a marker, how the ecology is, and then there is different molecules working together to change something. And then it's the components also of the bacteria maybe, so.
Can I follow on the question on the clinical trials? You mentioned, but you did not later on to discuss its effect on respiratory tract infections.
So we're still analyzing that. So in the trial 1, we basically did not see a difference in the infections and infestations as a system organ class, and we're still looking into the details and stratification in the trial to see what happens in the C-section born or those without siblings that have a little different risk. And this is what we showed in the first trial, where we see the effect on respiratory infections, that it was driven by the C-section born infants, and which was very high there, but which was luckily, let's say, lower in that last trial I showed you.
I cannot see whether there are questions from the audience because of the lights here. If not, I have a question which was already more or less raised by you, the comment, Norbert, regarding family size. Because you mentioned in your cohort, in your Finnish cohort, that it does matter so much. Please, you more elaborate on it.
Yes, thank you. So in our Helmi cohort, we indeed noticed that, especially in infants who were born by C-section and the colonization was delayed, when we looked for factors that compensated or helped them to be closer to the microbiota trajectory of the vaginally born, indeed having siblings became as a very important factor. And the hypothesis is, and we actually went back to the existing microbiota data, that there is a pool of infant-type microbes through the siblings, and we actually also saw that there is probably some sharing even probably back to the mother, because the mothers, if there were other smaller kids, so earlier siblings in the family, also had more these infant type of bifidobacteria. So it was really interesting, yeah.
Maybe just an addition, because we observed that in the second trial where we stratified now the microbiome analysis to siblings or no siblings, and we saw at baseline already those with siblings had higher bifidobacteria. And then we can compensate by giving the probiotic, synbiotic blend that we basically correct that it goes higher.
Thanks a lot. I do not see any more questions from the audience, and because there is one? No. Okay, I'm sorry, we have 60 minutes for this presentation, and I know Roberta is indicating, please have a look at the screen, scan the QR code to answer the survey. Please do it, because otherwise we will be punished. I would like to thank you very much for this participation in this symposium. Please, the take-home message and the only one is breast is best. I hope everyone agrees here, even if efforts are being made to narrow this gap between breastfed and formula-fed infants. Thanks a lot. Thank you very much. Thank you for the organizer, and thank you for your participation. Thanks, Anne, and thanks, Norbert. Thank you.